shoot, which one is actually recording? Okay, that. Something is... testing. Alright, that's confusing. Alright, hallo. En goedenavond. Ik heet Femmergene en ik ben terug met een andere stream. Hello, and good evening. This is Femmergene and I'm back with another stream. And I thought I'd do something different today. And I'm sorry that my stream is starting really late. But I'm only going to stream for, I think I'm going to stream for an hour and a half right now. And maybe come back later with something else. But I wanted to do a little bit of this just because it's just, I just like studying. Don't ask. I just like to study. So, okay, hold on. I have too many windows and tabs open. It's driving me crazy. Yeah. Too many windows and tabs open. I'm also streaming, I'm listening to music, but I can't stream music on Twitch, so it's being streamed to the Discord. You can listen to it there. So wait, what do I need? I need to see this. Yeah, and I need to see my stream manager. Where are you? Oh, there you are. I said too much going on. Hey, Rachel. That's sirens. You heard sirens? I didn't hear them. I have headphones on. I'm listening to music. How's it going? So let me switch the scene. Hopefully this works okay. Yes, you're alright. That's good. So here we go. Because you can't really see... Hold on. Let me see if I can... I gotta move this over a little bit. I need to find a better way to mount my camera somewhere. I think that's good enough. So how's your day? What have you been up to? Okay, that has to move. Okay, I'm like sorely running out of space. I need bigger space to do things like this, but reading and working. How's that going? Okay, so I'm actually on this part of this coloring book. Oh, in case people, okay, maybe I should explain what I'm doing. I'm totally spaced. So I have this thing called an anatomy coloring book. There it is, it's just back to here. That's this thing. It's called an anatomy coloring book. I've had it for years. I'm almost done it, and then got too distracted with life, with other life stuff. So, and I'm working through this section about the nervous system. And I also have, go back to this. I also have the physiology one, which was actually really fun too. I also have this one that I want to actually get further in. I think, what did I do? I did like the first 8. I did the first 8 for this. I definitely need to review what I what I read before. Can't really say I learned it if I don't remember it. How's the reading going? I have a ton of reading to do. Okay, hold on. Why is this crooked all of a sudden? That's better. Okay, that's better. I need a better way to mount my camera. I just don't have a better way right now. Anyways. So I'm working on the second section here, and this is about this plate is about the spinal cord. So the last couple I did, let's see. This whole section starts with nervous system, the organization of the nervous system. So you got the brain, and then you got the spine. You've got the spine here. So you've got the central nervous system in the brain. Why is it separate? Okay, this is the brain. This is the central nervous system, and then there's the peripheral nervous system. And gotta learn a little bit of the neurons, about the neurons, which obviously I have to review a ton of this stuff. Synapses and neurotransmitters. Oh, those synapses. Did you guys know that when you learn a new language, when you're learning a language, when you learn new things, your brain makes new synapses. So it is technically a physically painful process when you're learning, especially when it comes to languages, because you're learning new things that your brain has to make connections to, I think. Cerebral hemispheres. Yeah, brains. This was fun. I liked coloring this. Don't ask me why. And then tracks nuclei of cerebral hemispheres. So a little further in-depth look inside the brain. And yeah, I'm not entirely sure how to pronounce this. Encephalin? Diencephalin? I'm not entirely sure how to pronounce that. My student title says I'm not a medical professional or a student, so I have no idea how they pronounce that. Oh, the diencephalon is the smaller of two derivatives of the early forebrain. Fits between, but is not part of the surrounding cerebral hemispheres. See drawings 2 and 3. Yeah, okay. It's this thing here, this little thing here. Okay, and then the brain stem and your cerebellum, which is over here. There's your head, and then there's just part here, the lower part. Okay, and then, so now I'm working, and then we did that, I did that, and then the spinal cord. Yes, and now we're on the spinal cord. So I did the first half here. Well, I did this first half, basically, which is coloring some parts here and some parts over here. So now, let's color. I'm on around. Where is it? I read this earlier. Yeah. Here we go. Medium dark colors for E to I. They wanted bright colors for here, so medium dark colors. I don't want it. Okay, so posterior horn. I already colored the gray matter. I'm just double checking this to see what I did. D, A, B, C, D, yeah. I think this is all A, B, C, D, and this is all A, B, C, D, and this goes, so there's this thing, and then a close-up of this, over the cross-section. Posterior horn. Alright, so I did some of this already, but we're missing, oh, okay, E, F, G, okay, fine, G, H, I, so I need 5 medium dark colors. trying to think what colors to use. Because there's eye in the middle. And I don't want color for eye because it's in the center. I think I might just pull all these crayons, pencil crayons out. Makes it easier. So look guys, you're like, I always love this. I don't know if you guys can see. Got like 60 different colors. It's so helpful. Okay, maybe I should read the text here. The spinal cord consists of a central mass of gray matter, right, arranged into the form of an H. Yeah? Yeah, that's this whole thing, right? Yeah, you see it, it's like an H there. Into the form of an H and a peripheral array of white matter. Funiculi? Where is that? And a peripheral array of white matter. Yeah, do they have that in the picture? Or are we just supposed to know that? Brain, consisting of descending and ascending tracts. Okay. Oh, yeah, white matter is J. Duh, I'm stupid. White matter consists of KLM. Yeah. KLM. Yeah, that's white matter. That's what I was thinking. The amount of white matter decreases as the cord progresses distally, seen especially well in the sacrococcygeal region. Yeah, so the amount of white matter, so if you're at the top of your spine here, and there's white matter inside your spine, it decreases as the cord progresses distally. So as we're going further down, I think is what's going on. Seen especially well in the sacrococcygeal region. Yes, the sacrococcygeal region is down here, so there's less white matter. The gray posterior horns E, right? Well, it's technically gray, but we have to color it a different color so we know what section. Actually, columns, when seen in 3 dimensions, receive the central processes of sensory neurons and direct incoming impulses to the adjacent white matter for conduction to other cord levels or higher centers. Actually makes sense. The anterior horns, F. Posterior horn, anterior horns. The anterior horns include lower motor neurons that represent the final common pathway. That's in quotes, final common pathway for motor commands to muscle. Okay. Just the front and the back. So that's a cross section. What's the front and the back? Oh yeah, posterior is the front. Duh. Lateral horns exist only in the thoracic and upper lumbar cord. So lateral horns. Yeah, T1 to L2. T1 to L2. Trying to remember. There's L2 right there. Okay. Lateral horns exist only in the thoracic and upper lumbar cord and include autonomic motor neurons supplying smooth muscle in vessels and viscera and glands. So, autonomic motor neurons supplying smooth muscle and glands. Okay, the smooth muscle is in vessels and viscera. It is in the gray matter that spinal reflexes occur in conjunction with facilitatory and inhibitory influences from higher centers. Okay. Okay, interesting. Try to remember that. Anatomy, physiology, this is also factual. You have to remember everything. It's worse than languages. Okay, so I need medium dark colors. Let me see. What did I use already? Oh shoot. I did this so long ago I don't remember what color I used. What colors they are. I used to have a thing that told me what colors they are in here. Do I not have it? Hold on. I had a list of what colors my pencils are. Oh no, I don't have that list anymore. It's alright. We'll do it by sight. Okay. So is that one? That one. Orchid. I don't know if everyone can see. The lighting is probably the yellow lighting in my place is probably throwing off the colors. Yeah, let's do this one for eye. Do that one for eye, or should I do a blue one for eye? No, I can do that for medium dark colors, right? Medium dark. I don't know why I lost the list of the colors for my pencil crayons. I lost the list because that's how I remembered how to put the crayons back. All right, okay, people. Hey, Mike. Yes, I'm super organized. I put my pencil crayons back in a specific order. How's it going? How are you with colors, Mike? He seems good. Did you do your homework? Did you work on all your assignments? What color is this one? Okay, the reflection on these ain't helping me anymore. True blue. This is true blue. Oh no, I don't remember the order I put my colored pencils in, people. It's a tragedy. What the hell happened? Oh, it is here. There it is. Found it. See? This is the list of colored pencils I have. At uni, yeah. Good job, Mike. Okay, I need 4 more dark colors. What's this one? Well, medium dark, they said. Medium dark. So we want medium dark. What's medium dark? That's just a standard green. I mean, truthfully, depending on how hard you color, you can make it darker or not as dark. Is there a red, orange one? Do I have an orangey one? What's this one? Oh, is this that? That's just a red. Oh, no. Let's save the red for later. Red is always like to color blood. I don't know, I need 4 dark colors. I don't know what to use. Medium dark colors, that is. How about this one? Did I try this one? Cocoa. This cocoa. Cocoa color? Give me 2 more colors. Orange is not a very dark color. You know, with 60 colored crayons, you think I'd be able to find colors easier along the color spectrum, but they all seem to be like shades of the same thing. These are so light, like anything along the orange and pink is like really light, so I don't want to use them. This makes it so hard. Okay, what's this one? What's this green? Jade green. Jade green's fine. And don't I have a mauve color or something? Top? Is it taupe? Is it taupe? I think it's pronounced taupe. I don't remember anymore, people. Okay, we'll do that. I got 5 different colors. Sort of 5 different colors. Okay. Okay, E is the posterior horn. KLM. Oh yeah, I'm supposed to use light ones for KLM anyways. E is the posterior horn, so let's use this is orchid, and this one is, what's this one? Cocoa? Top. I use E, F, G, H, I. Oh, H and I are in the center. This is tricky. All right, let's just do. I want this for I actually. So let's do this one for E. Hope you guys are enjoying this. So I hope everyone had a good day. I think I need to sharpen this. Do you guys sell Crayola? Hello, good evening. Goedenavond, you know. Hoe gaat het met je? Please don't break on me. Okay. Okay, so E is a posterior horn, so I'm going to color this up. You know, it's been a long time since I've done this, so I might be doing this really poorly to start. Because I don't handwrite any. It's not doing very well. I need the edge of the pencil. This is not good. It's not sharp enough. How are you doing today? I am good. How are you? Let's see if I can. Let's see how much I actually get done in an hour and a half. Okay, is that sharper? I think that's sharper. How's it going? What did you do today, Chanel? When are you going on vacation? Okay, I'm gonna do this. You know, my vision is kind of bad too these days. Makes it harder to do things like this. is this pencil not? Oh, you know what? I should put chat on my phone. It makes it easier for me to see. I'm fine. I'm going on Saturday. It's cool. Where are you going again? I don't know if I asked you or if you told me last time. Oh yeah, Luxembourg. Bad me. I usually have a better memory, but I've been so distracted lately. I have so much stuff to do in real life that I just... Sometimes in real life just gets in the way. No problem. That's my excuse. Life in real life just gets in the way. Okay, I can barely see what I'm doing like this. Why? Could be the lighting in here. I need better lighting, I think. I'm gonna try something. Where's my flashlight? Whoa, that's so much better. At least for me, that is so much better, but probably not for you guys. I feel like this pencil's not sharp enough. I can't remember all the languages in other people's eyes. That's true, I guess. Depends. I mean, sometimes some days are better than others. I also can't remember all the anatomy either. I'm trying. I just like learning things. I think there's not enough time to... I think there's sadly not enough time to learn everything I want to learn. I want more time. 1,000 more years. Another 1,000 years. But I am listening to Russian music right now too at the same time. Like music in Russian. I think I can kind of see it okay without the light. The lighting in here is so yellow, it's hard to see, you know? But my phone is going to run out of battery if I do it this way the whole time. I know you guys can't really see that, can you? It's just this part's hard to see. The picture's easier to see. Alright, E. It's not too big, we just have to do these ones. But I gotta move the book around a bit. Okay, kind of hard to move my book around here. I have such small, limited space. Okay, here we go. So we have to color these. They wanted me to color this fairly dark, so I'm going to try that. Come on. Oh, wait, my phone is still doing this. What's my phone doing? I think I'm only going to just stream this for like an hour and a half, see how much I get done, maybe go play a game after. I'm actually kind of tired today, I think. I'm good, but I'm just a little tired, I think. Hopefully everyone can hear me okay. And the mic is right in front of me. There we go. Actually, should I move this camera? It looks a little lighter than the other one. Alright, that was E. And F. F should be this. Well, actually, no, H. These colors are too similar, maybe. Wait, where's G? It says lateral horn T1 L2 G. Wait, where's G? Oh, right there, I see it. I hope that works well. We'll see. Okay, F, anterior horn. Did I pick the wrong color? I don't know. I can always change the colors. Anterior horn. Actually, maybe I just do this one first. This one's sharp enough, I think. Can we be like more precise here? I guess some people use marker to do these, but I feel like if you used marker, it might go through the paper. Okay, so the anterior horn. Oh my god, I can't seem to control this pencil very well right now. By the the way, in case anyone's wondering what music I am listening to, it's being streamed in the Cute Terror Discord. Because I'd otherwise be falling asleep if I didn't have music to listen to while I did this, or if I wasn't watching a show. I guess I could put a watch party on. Oh god, why can't I get this to line up properly? Nate, these pencils are not cooperating.
Of course not.
Coloring. So that's the posterior horn, and we're going to do the anterior horn after. Closed captioning, you're funny. You never get me right. Why can't we have a multilingual closed captioning? Huh? Huh? I want to know. Oh my god, you know, it's been really a long time since I really did anything like with my hands like this. I can't even hold the pencil right anymore. That's so sad. You know, and then I had to write my signature on some stuff here when I got to the Netherlands, and I was like, man, I don't even remember how to write my signature. Good grief. I gotta admit, this kind of stuff makes learning fun, though. Actually, I'm stupid. If you guys want the music channel in Discord... Oh, dropping my pencil. How did I get that? there. Or rather, my studying music. That's it. Did I write study music? I meant my studying music. Music I listen to while I study. I don't know. Yeah, I really need like a lamp here or something, like a table lamp, I think. I think that's the problem. The overhead light isn't good enough. I think in the past, yeah, I think in Toronto I used a standing lamp that was right next to me. It made it easier because the overhead light comes from there and it's getting blocked. So if there was a light source next to me, it wouldn't really get blocked. Don't really have room for a standing lamp right now, though. I think I just realized why this is hard to do because I can't really see it. This overhead light. This isn't. Why does that one look darker than this one? Okay, well that's F. And G. G, I probably should use a really dark one. So the lateral horns, T1 to L2, okay. Yeah, I see G now. G is over here. You can barely see G. So this is G and it's pointing right there. That's G, lateral horn. Come on. Yeah, maybe I am going blind. Okay, colors. Why aren't you coloring? I should check this. Hold on. Why is that at 1080? Oh wow. Why is that even at? Just do that. Forgot to pause the stream in my stream manager. Okay, let's see. Why? Pencils? Why is it so hard to sharpen these? I want it super sharp. Ay, I think it broke. No, I'm trying to sharpen this and it just broke on me. No, that doesn't help. I don't know, maybe this is sharpened. Give me a sharp edge. A sharper edge of it. I swear I must really be going blind because I can't see very well. Okay, that actually might work okay. Damn it. Okay, where's my other pencil sharpener? Try this one. Oops. No, is that one better? Oh, this one might actually be pretty good. Maybe. Garbage bin. Let's see if that worked better. I'm glad you don't do this kind of stuff for years and then all of a sudden you can't do it anymore. Your hands just don't look the same. What the hell? Apparently all I can do is study languages and play games. Lateral horn. Alright. Okay, that was lateral horn over here. Intermediate zone is H. 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 Yeah, I'll do this one for H. What did they say about the intermediate zone? They didn't really say anything about the intermediate zone. No, why are you breaking on me? No, I hate when my pencils break. Oh jeez, it broke. Wanna suck it. That's annoying, it broke. Jeez. Things be flying. I hate when my pencils break. And it's not it's not a lot of coloring here, it's just that they break. Okay, I have to figure out where to buy colored pencils here in the Netherlands, but it's going to be sad because I won't be able to get the same colors. Oh my god, it broke again. No, stop breaking. Damn it. Damn it. Okay, this pencil's gonna go. No, come on. Right. Okay, please don't break again. Okay, let me read this passage again. The spinal cord consists of a central mass of gray matter arranged in the form of an H, and a peripheral array of white matter, funiculi, consisting of descending and ascending tracts. Okay, that might be sharp enough. The amount of white matter decreases as the cord progresses distally, seen especially well in the sacrococcygeal region. The gray posterior horns, actually columns when seen in 3 dimensions, receive the central processes of sensory neurons, recall plate 71, and direct incoming impulses to the adjacent white matter for conduction to other cord levels or higher centers. Plate 71 was just plate 71 was this, yeah, functional classification of neurons. And receive the central process. Yeah, because it's sensory neurons conduct impulses from sensory receptors to synapses in the central nervous system. The receptors may be sensitive to touch, pressure, pain, joint position, muscle tension, chemical concentration, light, or other mechanical stimulus, basically providing information on the external or internal environment and related changes. Sensory neurons are unipolar neurons with certain exceptions, bipolar neurons, and are characterized by peripheral processes, axons, cell bodies, and central processes, axons. Okay. Right. So that's relating, yeah. Okay, and the anterior horns include lower motor neurons that represent the final common pathways, quote, final common pathway, for motor commands to muscle. Lateral horns exist only in the thoracic and upper lumbar cord. The lateral horns are the ones over here, and they only exist in the thoracic and upper lumbar cord and include autonomic motor neurons supplying smooth muscle in vessels and viscera and glands. It is in the gray matter that spinal reflexes occur in conjunction with facilitatory and inhibitory influences from higher centers. Okay, I think I finally got this sharpened. I'm going to do this first then. H. Intermediate zone. Alright, that's that one. Let's see if I can actually color in the words a little better. It still feels like it's not sharp enough. Please don't break. I'm gonna just do this really gently. Come on. Oh, don't forget the challenge. Don't forget about the community challenge, people. It's still running, I believe. Thanks for the contribution, Rachel. I think we're at 69% raised, and we've got 5 days left. Come on, pencils. It just doesn't feel sharp enough, or is it just me? My vision's not sharp enough anymore. Oh my god, this is about to break. This is broken. This pencil is broken. It's a little broken. Oh no. Why is this one breaking? Oh jeez, do I have it? I have another one. I know I have another one in here. Come on. This is why I keep my old ones, even if I'm not. Maybe I'll switch it for the old one because this one's not working very well. Is it this one? Yeah, this one. This one just doesn't seem to be working very well right now. switch them out. Well, this is the right one. Yeah, this one doesn't seem broken. Okay, that's so much better. I gotta check, do they sell Crayola crayons? I need Crayola crayons. I need 60 of them. And you guys can see that. You love Crayola? Okay, they just sell them here in Europe. I also have actual, like, the wax crayons, and this is the sharpener for wax crayons, but I don't remember where I put my wax crayons. They're in a box somewhere. Hey, Adrenaline. Thanks for the contribution, Adrenaline and Mike. I guess I can just try to order it on Amazon and hope I get a good box of, like, not broken crayons. Pencil crayons. No problem. Okay, why is this one not dark enough? That just broke. But it might have broken a better way. Oh my god, stop breaking on me. Oh, come on. I don't like it when the pencil crayons don't cooperate. It's worse than a video game. It's rage at the pencil crayons for not working properly. Yeah, this one's broken for sure, because look, I can feel it, like this one just came out. I'm gonna put that, yeah, it's one of the newer ones, but I'm just gonna put that in the box for the older ones. I might actually just go and order some, see if I can order some more pencil crayons on Amazon or something, because you know. I'm pretty sure that's all the pencil crayons I have. I also have another set of artist pencil crayons, but they're not the same colors that I've been using for this, for the coloring book here. Yeah, I mean, technically I could study anatomy and physiology a lot faster if I wasn't coloring or everything. um, but I figured this would be fun. I mean, it is, generally. Except for the fact that my pencil crayon is not working right. Come on, am I blind? I'm blind, aren't I? I'm going blind. It's horrible. It's horrible. I went outside the line there. That's annoying. And I probably should order pencil sharpeners too. Well, this one's not breaking at least. Sometimes I feel like I should just take off my glasses and do this, and I could probably see better without my glasses on. I mean, technically these... Okay, I think this is working better now. Yeah. I feel like I'm seeing double. I'll try to fix these. Am I being picky just on something that I'm just using to learn? Yes, because I want it to look nice. It just makes it more fun to learn when something looks nice. That was a little bad there, whatever. Just let it go. Let it go. Let's try this. I'm gonna take off my glasses for a second. Let me just see something. Yeah, jeez. This one is so bad sometimes. It sucks to get older, you know, people. Yeah, so bad. I could see it. Alright, next one. Grey Commissure. Commissure? I'm not sure how to pronounce that. Grey Commissure. Okay, that's I. That one's easy. And this thing in the middle is a central canal that we don't have to color. Just double checking. Okay. Yeah, I'm pretty sure I left like a ton of old pencil crayons back in Toronto and just gave it to my nephews. It's fine. You know, they really should have made these letters bigger because, like, how sharp does my pencil have to be? Like, even like with my other ones that I did earlier, I was like, man, my pencil crayon has to be super sharp to do these. What did I just do? Probably also need to buy a new pencil sharpener. Let me see if that worked. Oh my god, this one's breaking. This one's breaking on me. I heard it crack, and it is actually breaking. Look. This one's orchid, right? Orchid. I'll try to find an older one. No, not that one. No, that's not the same one. It's maroon. Brown. I need new pencil crayons. Why do I still have this? Where are ya? Orchid color. Come on, I don't see you. That's maroon. Where are you? You usually can just match the colors. Come on. Wait. No, well, that's cocoa. Let's see it. That's not it. This one? Yes, orchid. Oh, broke. This one broke too. Well, look, this one actually looks pretty sharp still. I mean, you would think that the pencil crayons wouldn't get bad over a period of time. I mean, do they? I don't think they do. I think they just either made bad or they just weren't well made and then they crack. Okay, that one worked a little better. Alright, that's those. Now I'll put this new one in the old box. Can't believe it's like broke. It broke. Alright, so I need 3 light colors. Try KLM. KLM. Not use the same colors again. Yeah, light colors for K to M. What's this one? Turquoise? Turquoise are fairly light. Lime green. I always like lime green. Wait, no. What's this one again? What's this one again? Hazel. Hazel? I don't know if I want to use hazel. Do I want to use hazel? Tan. Hazel, tan. Sort of kind of like the brown spectrum. I guess I could. Hazel, tan, and then well, this is light green, lime green. Do you know how I get peach color? That's pink. Those are pretty dark. No. Which is this one? Which is this one? Can't read that. Man, why did you have to put like glowy letters and make it hard to read? And bad lighting. What the hell? Oh, it's salmon. Salmon colored. Do we want salmon color? Maybe M? M. Let's do salmon color for M. Because I feel like it. I don't think I used salmon color here already. Oops. I'm trying to hit the mic. Sorry, head bonking on the mic. I don't remember which way where I put these. Let's do hazel. I'll figure out where these pencil crayons go next. Okay, I'm gonna do hazel. Oh, I see something weird happened. Sorry, hold on a second. Let me just move this for a second. I'm gonna try not to close my laptop by accident. close. Oh, did I close my laptop? No, I didn't. Still here. Almost looked like I closed it. Hopefully not. Okay, that's not good. I can't sharpen this one. Okay, please don't break on me. Okay, so the white matter, so the gray matter, we just colored the gray matter, consists of the posterior horns, the anterior horns, the two lateral horns, so this is posterior is front, anterior is the back, lateral horns are along the side. Intermediate zone, right there is H. It's just part of this little H shape. And the gray commissure. So the white matter that I'm coloring next, white matter is right here, surrounds the gray matter. Consists of the posterior funiculus, the lateral, and the anterior. I might actually need a little bit more before I do the anatomy. Might not color the next plate on the physiology book. I might just review some stuff. It's not very sharp, but whatever. Posterior funiculus. Funiculus, maybe. Latin words, people. Latin words. So bright enough. Why? This is because I have an overhead light source instead of like, whoops, that's not right. I did not want to do that. I have an overhead light source, which isn't good. If I had a table lamp, it would be easier. I know it makes it hard for you guys to see because of the light casting glare there. This is so much better, actually. I think I do need like a table lamp or something, but I have no room for a table lamp. If there was a fun way to do this on the internet, I wonder if there's like websites that allow you to do this kind of way of studying anatomy. It would be fun. Somebody should create a site. Yeah, because without that light, like, I can barely see anything. Well, I can barely see anything. It's pretty dark. The lighting is pretty dark. The lighting is pretty dark in here. My hand is starting to get tired. It's been years since I've done this. Not sure how many years, actually. Years. Definitely years. I've been in the Netherlands for 4 years now, so and I was doing this before I moved, so it's definitely more than a few years. Okay, so K is posterior funiculus. I'll just put the flashlight away, don't need that for this next part. Alright, definitely still helps me a lot more to have the extra light. Did I move it too close? Is that better? Let me know if people can't see what I'm doing. Okay, you know what? I think I want the light source. Sadly, I don't have a like table lamp or something. No, this is hazel actually. Okay, that's the posterior funiculus. I don't know if everyone can see that very clearly, but right. Okay, the next one is, hold on. Next one is this one, lateral funiculus. And I guess we'll use the salmon here. We'll use the salmon, lateral. Okay, maybe I'll finish this plate in the next hour. So okay, it's not broken. Couldn't tell. Can you guys see what I'm doing? There's the light in the way. Yeah, it definitely helps having a better light source, honestly, for this. If I go too close, it's a glare, so I'll just do this. I can see, you guys can see. Man, I feel like I'm going cross-eyed. Hopefully not. Why does this feel like it's... my pencil crayon feels like it's... This one feels... it is broken. I was wondering. No, this one's broken too, look. Did I just get a bunch of duds in that one? Seriously. It was shaking. I could tell it was shaking. Okay, this was salmon. I need another salmon one because that one just is broken too. It's pink. I just hope. Oh my god, the other one is like this short. That's why I bought. That's why I bought a new set, right? It's. I mean, I could just try to sharpen it because it's not that short yet. But this one's fine. I had to buy another set, I think. Oh, I'm sleepy. Well, it's much better when it's not broken, you know? Okay, lateral funiculus. Funiculus? Funiculus? This is why Latin is good to study. Yeah, I do actually like doing this. It's kind of relaxing. I should really just do this at night before I go to sleep instead, because it's kind of relaxing. Well, there's also a bunch of other things I do before I go to sleep, so gotta make time for everything. It's hard, you know? Okay, lateral funiculus. That's the lateral funiculus. It's part of the white matter in your spine, and it surrounds the gray matter in the spine. Okay? And it's, since the gray matter is here, and this is the white matter, and then it's surrounded by, this blue one is the pia mater. Pia mater. Wait, is it pia mater? Did they miss a T? They just also call it pia in the text, so yeah, pia mater surrounds that, and then this thing here is the dura mater. Dura mater. Alma mater. Alright, so the last one is the anterior funiculus. So the anterior funiculus is over here. This is posterior, anterior, front, back, and the sides is lateral. anterior funiculus. This one doesn't seem broken. This one's lime green. It's fairly light, so it should work okay. Yeah, there's a lot in this book. And the last couple of sections are the male and female reproductive systems. So there's about, how many plates are there? 100 and something plates. 170 plates in the whole book. And obviously I've done some here. This one is like immune system, lymphoid system. So probably not the most efficient way to learn a subject, but kind of fun. I'm heading into 2 hours. I think broken pencils slowed me down. I might just go order some more pencil crayons. Yeah, it's fine. I think I definitely need a table lamp. You know, I could have just taken the table lamp I have in the bedroom. Just don't know if I could put it close enough, because I'd have to plug it in somewhere as well. Behind me. No, actually I have something else plugged in. Oh, it's just the fan, yeah. Alright, that's M, anterior funiculus, and that's this region here, inside the white matter. Why is it doing... I hate when that happens with the pencil crayon. Sometimes it's like inconsistent in how much color is. Yeah, so now it's just like, it's like the angle of the pencil is just messed up. It's not coming out very well, is it? I think I need new crayons. I hate spending so much money. I had to buy some new stuff this week already. Alright, that's the anterior funiculus right there. I think that came out okay. So that's a little bit of anatomy, everyone. Let me see if everyone. Wow, it really is hard to see that, huh? I don't know. Is that okay? Let's see if you guys. Hold on. It's pressing so hard to get some of the color into the thing that you can feel it through on the next page. Alright, so this one, this is the whole spinal cord here. That's what we're learning here. But this is what's inside your spinal cord. Gray matter and white matter. And gray matter is also in your brain, of course. Pretty sure. Yeah, white matter tracks. Hold on, where are we? There we go. I thought gray matter was also in your brain. What's wrong with me? Subcortical white matter. Okay, white matter tracks. Okay. And over here, there was, brain stem, the hindbrain, midbrain, cerebellum. Okay, spinal cord. I just looked that up. Gray matter. Oh, excuse me. I'm a little sick. No, it's just in the nervous system. It's not in the brain at all. Component of the central nervous system. Okay, so sorry. Yeah, it is in the brain. It is present in the brain, brainstem, and cerebellum, and present throughout the spinal cord. That's what I thought. Okay, about gray matter. I'm just going to read this article for a second, or skim this article. Gray matter is a major component of the central nervous system, consisting of neuronal cell bodies, neuropil dendrites, and unmyelinated axons, glial cells, astrocytes, and oligodendrocytes, synapses, and capillaries. It's distinguished from white matter in that it contains numerous cell bodies and relatively few myelinated axons, while white matter contains relatively few cell bodies and is composed chiefly of long-range myelinated axons. The color difference arises mainly from the whiteness of myelin. In living tissue, gray matter actually has a very light gray color with yellowish or pinkish hues, which come from capillary blood vessels and neuronal cell bodies. The function of gray matter. Gray matter consists most of the brain's neuronal cell bodies. The gray matter includes regions of the brain involved in muscle control and sensory perception, such as seeing and hearing, memory, emotions, speech, decision-making, and self-control. The gray matter in the spinal cord is split into 3 gray columns. The anterior gray column contains motor neurons. Right, yeah. These synapses with interneurons and the axons of cells that have traveled down the pyramidal tract. These cells are responsible for the movement of muscles. That's what I was reading in the book, yes. The posterior gray column contains the points where sensory neurons synapse. These receive sensory information from the body, including fine touch, proprioception, and vibration. This information is sent from receptors of the skin, bones, and joints through sensory neurons, whose cell bodies lie in the dorsal root ganglion. This information is then transmitted in axons up the spinal cord in spinal tracts, including the dorsal column medial lemniscus tract and the spinal thalamic tract. And there's links there on the Wikipedia article for those other things about the different tracts. The lateral gray column is the 3rd column of the spinal cord. The gray matter of the spinal column can be divided into different layers called rexed laminae. These describe, in general, the purpose of the cells within the gray matter of the spinal cord at a particular location. Interesting. I could have probably put that on the I could just probably have done that. Oh no, it's not the right size, is it? Let's see if I can get this the right size. Oh, I can't see what I'm doing. Hold on a second. Don't know if that's the right size. I didn't set that up. I didn't think to set up the display capture. Some other time then. So, where am I? Alright. So maybe if I'm... Actually, I'll switch it to this. I got an idea. Switch it to this. I'll just hold this up for people to see. Maybe you guys can see better this way. I don't know. So let's see. I hope you guys can see that better. Or should I use the side camera? I don't know if that's better or not. Kind of hard to see, isn't it? So the bottom part is the one I just finished coloring. Is the bottom part right there. Okay, that's a cross section of your spinal cord. Actually, okay, maybe I will try with the other one. Hold on. Let's try this. Just move it slowly, carefully. Okay, I have some things on top of the cord here. I don't know if you guys can see or not. Will this work properly? If I do this? Okay, this is the section I just did. This is this section here. The gray matter and white matter, right? And gray matter is this thing here. That's the gray matter. And then, let's see if I zoom in on that. Hopefully, I don't know if it's clear. And the white matter is this area surrounding the gray matter. Okay. Let's put this back to where it was. Where's my camera going? I think I need to move this. Trying to get this camera back to where it's supposed to be. There you go. Is that's better. There we go, everyone. So that is a little bit about the anatomy of the spinal cord. I'm gonna put this away for now. The next thing I need to do, I don't know if I'm gonna be streaming it next time. We'll see. It's the problem of not having good lighting. This is actually like, this book is pretty old. This is actually like tearing off. The plastic on the cover is actually coming off. That's ridiculous. Wait, when did they? This is the 3rd edition of the anatomy coloring book. So when did I buy this? Oh my god, really? Well, I don't think that's when I don't think that's the year I bought it, but this version, this edition, it's the 3rd edition of the anatomy coloring book. It's this one, the anatomy coloring book by Win Capit. It's the 3rd edition. 3rd edition was published in 2002. I must have bought this in like, I want to say I bought this maybe 2007, 2008, and just been like, well, but to be fair, when I first bought it, I did a lot at the beginning, and then I got distracted, heavily distracted by this over the years. Distracted from it over the years. But I've done pretty much. The only thing I haven't done in this is the nervous system, central nervous system. Everything related to the nervous system, I haven't done that yet. And I haven't done the rest, the reproductive systems. Alright, so I'm going to pull out the physiology coloring book, which is probably newer. It's also by Capit. I don't know if you guys can see this very well. This is also by Capit. You guys can see that? This one's also by Capit. Somehow it got warped. Why is this so warped? Probably when I shipped my stuff over from Canada to the Netherlands. Anyways, I think I won't color the next plate tonight. I'm gonna quickly see if I can review things in here. This is the second edition. Second edition of the physiology coloring book. This was actually published even older, 2000, but I'm pretty sure I didn't buy it in 2000. So the first unit is cell physiology. Let me see if I can refresh my brain. I don't know how many times I read this and just forget everything. I like anatomy and physiology. I like this stuff, but it's like a lot of this stuff is so factual, it's hard to remember everything unless you're actually working in the field, in the industry. So as you can see, I've already covered this plate. Okay, DNA replication. So the first unit, the first plate was cell structure. Just cell structure. How did we color this? Okay, the cell membrane. Cell membrane, the cell plasma membrane is this thing, this color right there. There's the cell plasma membrane, right? And the nucleus. The nucleus is over here. Yeah, this is the nucleus. This whole thing's the nucleus, I believe. Let's see. Okay. Let's go. Epithelial cells, that's like skin cells. DNA replication and cell division. So DNA replication, it's all. Okay, let me see. Cell division first and DNA replication. This part's interesting. In cell division, one parent cell divides into two daughter cells to create new cells. Although some characteristics, example weight, of the daughters may be different from the parent, they are identical in the most important way. They both carry the same fundamental set of genetic instructions that govern their activities and reproduction. This instruction set, the genetic code, is provided by the detailed structure of DNA. Deoxyribonucleic acid, DNA, molecules that are packaged within the cell nucleus. Yes, so DNA is within the cell nucleus, right? We have the nucleus here, right, but they don't, they're just not really showing the DNA. There's the cell nucleus. And DNA is inside there. The most prominent cellular organelle, the nucleus, contains genetic material, genes, DNA, and chromosomes. Information stored in genes is utilized in everyday cell life and reproduction. The nucleus also contains a smaller body, the nucleolus, that consists of densely packed chromosome regions together with some protein and some RNA strands. The nucleolus initiates the formation of ribosomes, structures that are required for protein synthesis. The nucleus is surrounded by a double membrane that is riddled with pores involved in transporting materials between the nucleus and the rest of the cell. Okay. My brain is imagining what's going on in the body. So three phases. Yes. Okay, 3 phases. So 3 phases of cell division. We've got interphase, mitosis, and cytokinesis. Right. Replication of these molecules and their distribution to each daughter cell ensures the continuity of cell characteristics with each division. Processes involved in the cell cycle takes place in 3 phases. Interphase. The cell increases in mass. This occurs through the synthesis of a diversity of molecules, including an exact copy of its DNA. This part. I don't know, people can see that very well. That portion of molecules, including an exact... hold on. Jeez, I misread the line. This occurs through the synthesis of a diversity of molecules. I go back to the beginning. Interphase. 1. Interphase. The first phase. Interphase. The cell increases in mass. This occurs through the synthesis of a diversity of molecules, including an exact copy of its DNA. That portion of interphase in which DNA synthesis takes place is called the S period. Okay, it is preceded and followed by 2 gap periods called G1 and G2 respectively. See illustration. Which part of the illustration? I'm not sure it's in... Wait, what? Oh, phases of the cycle. There we go. Here it is. So, phases of the cycle, right? Okay, the portion, this is the phase, yes, that portion of interphase in which DNA synthesis takes place is called the S period. This is the S period. So DNA synthesis takes place there. And it's preceded by G1, a gap period, and G2, a second gap period. During the S period, the centrosomes are also duplicate. During the S period, the centrosomes also duplicate. Okay, so here, over here, we've got the interface over here is a drawing here. I guess it's hard for everyone to see that. So here, let me just see if I can do this quickly. See if oh, jeez, did not mean to move that. So we've got phases of the cells, phases of the cell cycle. So they're saying the S period is where DNA synthesis takes place. And there's a gap period before and a gap period after. So G1, G2, right? And then the second phase is mitosis. The third phase is cytokinesis. So interface over here, interface right here. I don't know if everyone can see that very well. It says, during interphase 1, uncoiled DNA contained in chromatin replicates. 2, following replication, DNA becomes active in directing the RNA and protein synthesis required for cell division. DNA becomes active in directing the RNA and protein synthesis required for cell division. The centrosomes duplicate. So we've got, let's look at the picture here. We've got, okay, the plasma membrane is this area right here. Nice little border. And then the nuclear, the blue area, the blue circle here is the nuclear envelope. And the chromatin is inside. This squiggly stuff inside is chromatin. Chromosome 4 to 6, is that in there? No. But in here we've also got centrosome. And what's this one? Is that? No, that's just gray. Alright. Next, mitosis. The next phase is mitosis. This part's fascinating to me. Mitosis. DNA is replicated and moved. Following the second gap, G2, the cell enters mitosis, a stage in which the replicate sets of DNA are bundled off to opposite ends of the cell in preparation for the final stages, in which the cell splits in two. Follow the diagrams in the plate for details. Yeah. This one. Yeah, DNA replication, and then it splits into two. Mitosis begins when DNA molecules which had been unwound during interphase become highly coiled and condense into rod-shaped bodies known as chromosomes. Okay? At this stage, what are we talking about here? Mitosis. Yeah, okay, mitosis, right. At this stage, each chromosome is split longitudinally into two identical halves called chromatids. That's over here, I think. That's what we're looking at. Each chromatid contains a copy of the duplicated DNA along with some protein that provides a scaffold for the long DNA molecules and helps regulate DNA activity. Meanwhile, the nuclear envelope begins to degenerate, and outside the nucleus, centrosomes migrate to opposite ends of the cell to form an elaborate structure of microtubules called a spindle. Each chromosome attached to these microtubules lines up at the cell's equator in such a way that its two chromatids are attached to microtubules leading to opposite ends of the cell. The microtubules then pull on the chromatids, moving a complete set to opposite parts of the cell. Finally, the chromatids at both ends of the cell begin to unwind and become indistinct while a new nuclear envelope forms around each of the two sets of chromatids. Not sure closed captioning got all that. So mitosis, the picture over here I heard something. Mitosis, prophase. During prophase, the nuclear envelope begins to break down. Yeah, that's this thing. You break down little dot things. The two copies of DNA begin to coil and supercoil, forming chromosomes. Yes, that would be this thing here. See the long things? Centrosomes separate, migrating toward the cell poles located at the extreme right and left in the illustration. The chromosomes organize microtubules which form the mitotic apparatus. And then metaphase. During metaphase 1, the nuclear envelope and the nucleolus disappear. 2, the chromosomes line up around the cell equator, imaginary line connecting the top and bottoms in the illustration. Okay. Anaphase. During anaphase, microtubules attach to proteins, kinetochore, that are bound to a constricted region, centromere, of the chromatid. The sister chromatids are pulled to opposite poles of the cell by microtubules of the spindle. Don't know if you guys can see this very well. Telophase. During telophase, a new nuclear envelope forms around the chromosomes, near each of the two poles as two nuclei and two nucleoli begin to appear. The chromosomes uncoil, forming chromatin. The spindle fibers disappear. And then the third phase. Third phase. Is cytokinesis. The cell divides. This is the final stage. Cytoplasm division takes place as a furrow develops, becoming deeper and deeper until the original cell is pinched in two and the daughter nuclei formed during mitosis are enclosed in separate cells. At this point, the daughter cells enter the G1 stage of interphase, completing the cycle. During cytokinesis, the daughter cells separate. A furrow, narrowing, forms along the equator and progressively constricts the cell until it separates in two. First signs of a furrow can be seen as early as anaphase. And then the next part is DNA replication. So, mitosis. Let's show you guys the mitosis diagrams. Let's show everyone the mitosis diagrams. Alright, so we've got, I don't know if I got this angled right. There we go. Mitosis. This whole thing's mitosis, and this is prophase of mitosis. And then metaphase. And then anaphase for here. And then telophase. And after mitosis, after mitosis is completed, you have cytokinesis down here. That's cytokinesis. So the cells have, now it splits off into two daughter cells. And then, so DNA replication, I'm not going to read the whole thing about DNA replication out loud actually. It's pretty long. Let's look at DNA replication. So DNA is located in the nucleus of the cell, right? This is the cell, and this is the nucleus of the cell. And your DNA strand is located inside the nucleus of your cell, and also inside. Probably looks like it's inside chromatin. Right? Is that what we read? I don't remember now. I'm refreshing my memory. So DNA, sorry, I know you guys. So DNA replication. Can you say this very well? Let's see. Oops, sorry about that. Bang, crash, bang, boom. DNA replication. So there's a DNA strand here, the double helix, right? But then eventually it splits off, right? This is the original strand, but then it splits off. Everyone can see that. So DNA strand right here, and it splits off into two. And so looking at that, while I read this, replication is the process in which an exact copy of the DNA molecule is made. The double-stranded DNA in the chromatin, it is in chromatin, unfolds, separating at sites where the two strands are attached, hydrogen bonding sites between the complementary bases adenine, thymine, guanine, and cytosine. Each strand consists of a backbone, leg of the latter, plus attached bases, added by enzymes such as DNA polymerase, and using the original separated strands as a template, two new strands are synthesized as nucleotide building blocks, molecules containing the base together with the backbone materials, sugar and phosphate, are attached to the template. An exact copy is always obtained because complementary bases are the only ones that are attached. An adenine will attach only to a thymine and vice versa. A guanine attaches only to a cytosine and vice versa. Oh. Okay, gotta read that over myself at some point. Yep, that wasn't supposed to happen, camera. Ignore that, that just went all the way up, didn't it? That's actually funny. Stay, what the hell? And the camera is being funny with me. What's this one? What was that? That was, thinking. That's K. DNA polymerase. Alright. So the next thing was DNA expression and protein synthesis. Right? It's understanding how DNA works. Then metabolism. Then we learned a little bit about metabolism, role and production of ATP. ATP is... What is ATP again? Yeah, like I said, I have to review this because it's all interesting to me, but so much factual stuff. Metabolism, respiration, and the citric acid cycle. See, now we're getting into stuff that's actually kind of important for for fitness, from a health and fitness perspective. Metabolism, respiration, and the citric acid cycle. Okay, the structure of cell membranes. Okay, that's a little more just like anatomy. Solute and water movements. Again, these things are actually really important. Okay, so the next thing I needed to do was pathways for membrane transport. See, well, my brain is going, wait, I don't remember anything. So I need to review some of this on my own at some point. And two identical daughter cells. Oh, you like that part, right? Notice they didn't say son cells, they said daughter cells. It's kind of funny. Alright, I'm not going to start coloring this part. I need to review what I already learned in this part, in this whole thing. Every time I review it, my head still goes, what? I don't remember anything. Don't know why. Maybe too many languages makes it hard to remember. I think it was 2.5 hours. That was longer than I wanted to do. Doesn't help when your pencil crayons break. Really doesn't. Alright. Let's see about rating somebody. I might just I think I'm just gonna take a break. Because I'm tired today for some reason. I'm in a good mood, I'm just tired. I don't like being tired. Might come back with just a brainless game. Okay, what's going on Twitch tonight? What is going on Twitch tonight? Oh, we can do that. Here, let's go. Wait, who's studying? Let me see what this is. I think I probably have to stop playing my music. Yeah, okay, so I'm gonna quit the music channel on the Discord, guys. But that's done now. And then I'm gonna stop my music. Or at least pause it. Where's my music? Alright, makes it easier. Someone's doing Duolingo. I don't know what they're doing though. Do you guys want languages or some games? I think I'm in the mood for a game now. Right. Someone's doing Spanish, I guess. I don't even remember following this person. How bad is that? You know, I might send you over to a chill streamer who's playing Final Fantasy 14. I think that's what I might do. No, let me see. I think Doshino is just here. Let me peek in on the person playing Final Fantasy 14 right now. Oh, you know what? I don't hear anything because I had to mute my Chromebook playback. There we go. Okay, don't go away, everyone. Is it yet? He's still doing... Okay, well, here's what I'm gonna do. I'm gonna do a shout out for Shino because he is streaming Final Fantasy 3 Pixel Remastered right now. Shino's a native Dutch speaker, and he streams in Dutch. You can check that out if you want, if you're feeling ambitious about watching Final Fantasy 3 Pixel Remaster in Dutch. And I'm going to send you over to somebody playing Final Fantasy 14. So I'm going to just send a small little Ninja Bot raid over to Mighty Mar, who's playing Final Fantasy 14 online, and I believe he's playing the newest... Well, I don't think the latest expansion isn't out yet. He's playing Shadowbringers, which is the last one that was released. The new one... Wait, actually, is Shadowbringers the newest one? I lost track, people, because I'm only at the beginning of Final Fantasy 14. Yeah, Shadowbringers is the third expansion. There's going to be a fourth one, I believe. I don't remember what the fourth one's called. What's the fourth expansion called? I don't know. Oh, Endwalker. Yeah, Endwalker's not released yet, as far as I can tell. Endwalker is on November 23rd. Yeah, Endwalker releases on November 23rd. But anyways, I'm gonna do this. I'm gonna do this raid Mighty Mar, and I'm gonna say see you all later. Thanks for watching this little random stream, a little bit of anatomy and physiology. It's just been subjects that I've been interested in for years and haven't had much time to continue with them. So I hope you enjoyed watching. Thanks for watching. Bedankt voor het kijken. Tot de volgende keer. See you next time. Or maybe later. I might pop on later. I don't know. But I will be back tomorrow for sure. Tomorrow evening with more Mass Effect 2 Legendary Edition. Anyways, so that's it right now.